Hello everyone. Today I'll be explaining genetic engineering and the techniques used to apply it. Traditionally, humans have manipulated genomes indirectly by controlling breeding and selecting offspring with desired traits. However, genetic engineering involves the direct manipulation of one or more genes. One gene that produces a desirable trait is inserted from one organism into a different organism. So, in simpler terms, it is the changing of an organism's DNA to give the organism new traits. Genetic engineering is possible because the genetic code is universal and is shared by all organisms. Genetic engineering is based on the use of recombinant DNA technology. Recombinant DNA is DNA that contains genes from more than one organism. Bacteria are commonly used in genetic engineering to produce recombinant DNA. One reason is because they contain plasmids, which are closed loops of DNA that are separate from bacterial chromosomes and replicate on their own within the cell. So how does the process happen? First, a gene with a desirable trait is picked and is extracted by cutting the DNA using restriction enzymes. Then, the bacterial plasmid is also extracted and cut using the same restriction enzymes. The sticky ends of the plasmid and the foreign gene match and they bond, forming recombinant DNA, which is inserted again into the bacteria where the new trait will be expressed. Scientists are trying to use recombinant DNA in several different ways. Recombinant DNA technology has helped us overcome many issues, some of which are health, agriculture, and environmental issues. I'll be talking about each one of them separately. Recombinant DNA technology is playing a vital role in improving health conditions by developing new vaccines and pharmaceuticals. One famous example is the production of insulin using genetic engineering. The process is really important because it has helped provide a source of insulin for diabetics to lower their blood sugar levels. Cow insulin was used in the past, but some people were allergic to it. However, human insulin produced by using genetically engineered bacteria does not trigger an allergic reaction. Another benefit is that the insulin is acceptable to people with a range of religious beliefs who may not be allowed to use insulin from animals such as cows or pigs. The product is also very pure, and the human insulin can be made on a commercial scale, reducing costs. Genetic engineering has allowed scientists to give plants new traits such as resistance to frost, diseases, and insects. For example, a gene called Bt is introduced into plants to allow them to make their own natural pesticide, reducing the amount of chemical pesticides needed to protect the plants from insects. These crops, called GM or genetically modified crops, have helped increase crop yields so more food is produced more quickly and cheaply. Genetic engineering strategies have been employed to tackle environmental issues such as converting waste into biofuels and bioethanol, cleaning the oil spills, carbon and other toxic wastes, and detecting arsenic and other contaminants in drinking water. Concerns about genetic engineering. There are concerns about possible effects of genetically engineered organisms on both human health and the environment. And at an even more basic level, some people wonder whether genetic engineering